Hello everyone. Let's revise some of the important topics from chapter Principles of Inheritance and Variation. So in this video, we will revise monohybrid cross. So now what is a monohybrid cross? As the word suggests, mono means we will consider a single character or a pair of contrasting traits while making the cross. So, as you have studied already that Mendel took 7 different characters in his experiment. At a time, now we will focus on only one character and analyze how it is inherited through various generations. So, we will take the example from textbook itself. Here you can see we have considered a single character only, that is plant height in pea plants. Okay, you can either say a single character is considered or you can say a pair of contrasting traits, right? If you are saying pair, then you have to use the word contrasting because plant height in pea plants exist in two contrasting forms, that is tall and dwarf, that is short plant. So now let's see what happens if you make a cross between a tall and dwarf plants. Also, we have to remember that when we start with any like monohybrid or dihybrid cross, the parental generation which we take need to be a true breeding or pure line. So these tall and dwarf plants which have been taken here in the parental generation, these are obtained by repetitive self-pollination, and if those plants showed stable inheritance, that is if we let's say take a tall plant, do self-pollination, and in the next generation we get all tall plants, then again do self-pollination, we get tall plants, and so on for several generations. If same character is expressed in all the plants, then it is a pure line or a true breeding line. Same applies for a separate plant population which will show the short height, right? So take the pea plant with short height, carry out self-pollination, look out for the next generation, right? They should have short height. Next generation, short height. All plants in all generations will have short height. That means again that population is true breeding or pure. So in such a manner, the true breeding lines are obtained. One population shows true breeding tall plants and the other one is true breeding dwarf, and these plants will be used in the parental generation. So here we will start with the monohybrid cross. So this is the parental generation. We have taken plants with contrasting characters. One is pure tall, as you can see as it is tall, it is represented diagrammatically also, and the other one is short. Okay, let's make a cross. Now when he crossed the tall and dwarf plant, usually we would expect we would get maybe a medium height plant, but it is not so. Mendel observed that all the plants in the first generation, right, let's say he got 1000 plants, then all the 1000 plants were tall in height. This generation here is called F1 generation. You can see F1 means first filial generation. Okay, all the offsprings in F1 generation were tall. The next step was selfing, that means self-pollination of tall plant. Okay, that is the F1 generation plants. So here we have shown the selfing. Okay, self-pollination is done. Now what will be our expectation if we take a tall plant and do self-pollination? We will expect that all the next generation offspring should be tall, right? As the parent is tall, the offspring should be also tall, but it was not so. In the next generation, that is the second generation of offsprings, that is F2, second filial generation, it was observed that about 75% of the plants were tall and the remaining 25% were dwarf. As you can see here, 4 plants are shown in the F2 generation, out of which 3 are tall and 1 is short. So now the question is, how come there is a short or dwarf plant if the parent was tall only and we did self-pollination in that tall plant, right? This is also a question. And Mendel thought about it, like how to solve this problem, how we can explain this. And then Mendel came with an idea that there is something in the plant, in the pea plant, that is controlling the characters, right? Tall and short height. But what was it? That was not known when Mendel was doing the experiment, right? In the year like 1856 to 1863. The experiments were long back, and that time we had no idea about what is DNA, what is its structure, what are genes, chromosomes. Now we know that characters are controlled by genes, but that time no one had any idea about what are genes, what is DNA. So it was a question, what is actually controlling the characters, right? How tall and short height actually is controlled. So Mendel simply used the word factors to represent the thing which is controlling the characters. And he simply said that, as we can see, if we cross tall and dwarf plants in the parental generation, and we are having only tall heighted plants in the F1, so this height is dominating, right? So we use the letter T, capital T for tall height, and for dwarf plants we use the letter small t. Okay, so the character which was expressed here in F1, this is called dominant, and the character which was not expressed in F1 but we saw it in F2, that is dwarf, this is called as recessive, right? Now we need to explain how we can show the inheritance of all these factors. Okay, and now with further research we know these factors are actually genes. Okay, so how we can write this cross? Okay, this is also given in your textbook. So here the same cross is shown, but now we are representing the genes, the factors also in each generation. So you can see tall and dwarf plant, right? So as we started with pure line or true breeding line, that is why we have mentioned it like this, capital T, and here for dwarf plant small t. The factors occurs in pairs. Okay, this was also told by Mendel that in the plants, in the parents, the factors occur in pairs, and now we know it is true because our chromosomes are also present in pairs, the genes are also present in pairs. So that is why we have written it in pairs. Now, when gametes will be produced in the plant, obviously when plant will produce gametes, then in between we will have meiosis, right? Meiotic division, and the genes, the factors will separate. Now in this case, as you can see here, we have capital T. If we separate capital T, then both the gametes, right? Like 50% will have the first capital T, the remaining 50% will have the second capital T. So it is mentioned twice, but technically we have only one type of gamete containing capital T. Okay, and in the dwarf plant also both the small t will separate because of meiosis. Meiosis is reductional division, so these factors will separate, and like we can see, whatever gametes are produced in this dwarf plant, they have a single copy of small t, right? So this actually explains the law of segregation, that during gamete formation this pair of factors actually separate. Okay, now either this capital T will fuse with this small t or with this small t, or the second capital T will fuse with this small t or this small t. Whatever the combination is, you can see all the plants in F1 generation, they have genetic makeup capital T, small t. You can see all these four plants, they have the same genetic makeup capital T, small t, and the height here is tall, right? All the F1 generation plants are tall. That means tall height is dominant. But we can see the genetic makeup is hybrid, that is heterozygous, capital T, small t. That means the character tall height was dominant. So we simply say that capital T is the dominant allele and small t is recessive. Though both capital T and small t are present, only capital T is expressed. That is why tall height is dominant. Next step, what do? We do the selfing of F1 plants, right? So these hybrids which are present in the F1 generation with capital T small t genotype, these are self-pollination. That is why we have mentioned it like this, capital T small t crossed with capital T small t. Now again, when gametes will form in these F1 plants, the capital T and small t will separate, and now you have two types of gametes, 50% with capital T and the remaining 50% with small t. Let's say if these are male gametes, then these are the female gametes. Again, 50% with capital T and 50% with small t. Now let's see the different combinations possible, okay, of offsprings. Now this is a very easier way to represent the possible gametes and also all the possible offsprings. This was given by Reginald C. Punnett, okay, and this is called Punnett square. This is a very easy representation to show all the gametes and possible offsprings. Now let's see what are the offsprings. If this capital T and this capital T, these gametes fuse, you have the offspring capital T. As both are dominant forms, you will have tall height. Now let's see this. If capital T fuses with this small t, okay, then we have this plant which is appearance wise tall, but what is the genetic makeup? Capital T small t, hybrid, right? Similarly, this one is also tall but hybrid, and if this small t and small t fuse, you will get the genotype small t and dwarf height of the plant. So here you can see in the F2 generation, the second filial generation, 3 plants are tall and this one plant is short. Okay, the appearance which you see, like for example, you can see the height, right, whether the plant is tall or dwarf. Like in this case here, you can see it's written the F1 generation plant is tall. So whatever you can see is the phenotype. Whatever you see is phenotype, and the genetic makeup of that organism, like we have mentioned here, right? Capital T small t, whatever we write, capital T small t or small t, that is genotype. Okay, so in F2 generation, we can see phenotype wise there are 3 tall plants and 1 dwarf plant. And genotype wise, what can we see? There is one plant which is homozygous, that is pure tall, right? Then there are 2 plants which are hybrid, heterozygous, tall, and this one last plant is dwarf. And genetically, right, genotypic ratio means we have to see the genetic makeup, right? There is only one plant with small t. So in the F2 generation, we can write the ratios as first phenotypic ratio. This is for F2 generation, okay? Phenotypic ratio, okay? We have tall and dwarf plants. We have 3 tall and 1 dwarf plant, so the ratio is 3:1. And the genotypic ratio, where we will see only the genetic constitution, genotypic ratio. Here we have 3 types of genotypes, capital T, capital T small t, and small t, right? And this comes in ratio 1:2:1. So you have to remember these ratios. The phenotypic ratio, F2 generation plants of monohybrid cross, okay, the ratio is 3:1, dominant is to recessive, and genotypic ratio is 1:2:1. Okay, now there is only one question, okay? We can see only the phenotype, whether the plant is tall or dwarf. How come we know that this tall plant is capital T and these 2 plants are capital T small t? We can't see genes, right? We can't see the factors. Then how do we know that these 3 plants, okay, out of which this first one is homozygous and these 2 are heterozygous? Actually, what we can do is, if we have plants with unknown genotype, see, for recessive it's not a problem because we will have a fixed genotype, small t, but for dominant character, now it's a question whether it is homozygous dominant or heterozygous. So in this case, what we can do is, we can take this plant whose genotype is unknown and we can simply cross it back with the recessive parent, that is small t, okay? Let's quickly see what we can do. See, we have 2 questions here. Either our plant with unknown genotype is capital T, or it is capital T, small t. We don't know what it is, okay? And we are crossing it back with the recessive parent only, okay? That is small t in both cases, okay? Now here in this first case, we can see capital T, both will separate, law of segregation, and whatever capital T you take, you will get only one type of gamete. Similarly, in the dwarf plant, small t, you will get only one type of gamete, right? Both these gametes will fuse and you will get the offsprings. All the offsprings have capital T, small t genotype, and appearance wise they will be tall. So you can see all the offsprings obtained in this cross will be tall. Okay, so based on this result, you can tell that the unknown genotype was capital T. Now, what if the unknown plant in the F2 generation or whatever, F1 generation also, how do we know it is hybrid genotypically? What we do? We take the plant, cross it with recessive parent. Now, let's write the gametes. For this plant, capital T, small t, we will have two types of gametes because of the law of segregation. Both the alleles, both the factors will separate. You will have capital T in 50% gametes and small t in the remaining 50% gametes. In the recessive, you will have only one type of gamete, that is small t. Either these two gametes will fuse or these two gametes will fuse, and you have the offsprings 50% capital T, small t, and the remaining 50% are small t. So these first 50%, these are appearance wise, means phenotypically tall, and the remaining 50% will be dwarf or short heighted, and these will be equal in ratio 1:1. So simple. If the unknown genotype was homozygous, like in the first case, capital T, then by doing this cross with the recessive parent, okay, this is called as test cross. What is this called? Test cross. Okay, very important, test cross. Okay, all the offsprings here were tall. Okay, this tells us that the unknown genotype was capital T. And if the unknown genotype was capital T, small t, that is hybrid, then the offsprings will show ratio 1:1. Okay, so with the help of such test crosses, we can easily find out the genotype of unknown plants. I hope this video was helpful for you, both monohybrid cross and test cross. Let's see the other topics like incomplete dominance, codominance, linkage in upcoming videos. Thank you.